Okay, ladies, so as we continue our discussion on the chemistry of life, we just finished properties of water, and now we're ready to talk about macromolecules, carbon-based molecules. So we're going to be talking about 4 macromolecules. And to begin our discussion, before we get into each of the macromolecules, it's important that we talk about carbon. We are carbon-based life forms, and carbon is the backbone of every single one of these macromolecules. So carbon is super unique because of the way that it bonds. And what makes it unique is the fact that it can form covalent bonds, which we know that means they are sharing electrons. We know that it can bond with 4 other atoms. So when I look at any single carbon, so let's look at this one down here. When I look at this carbon, I can see that it has 1, 2, 3, 4 bonds attached to it, to 4 other atoms. It just so happens that all 4 of the, well, not all 4, 3 of the atoms are hydrogens, 1, 2, 3, and the 4th atom it's bonded to is carbon. So once again, remember, a covalent bond means sharing electrons, and it also means that we use a solid line to indicate that these 2 atoms are sharing electrons. Now, in a carbon-hydrogen bond, this type of bond shares electrons equally. So I'm going to put shares electrons fairly equally. Now, it may not be exactly, because to be exactly equal, they have to have the same electronegativity. And they don't have the same electronegativity. But the difference in electronegativity isn't super great. It's not a large difference. Therefore, we would say it's fairly equal. So if there's a fairly equal sharing of electrons, then there are no charges. There's no positive, no negative charge. And if there are no charges, what does that mean? That means that the molecule is not polar. In other words, we say non-polar. So carbon-based molecules, which are super unique and have covalent bonds, they can form 4 bonds. They do so in 3 general types of structures. So let's look at each of these. They can be a straight chain. So right here I have a straight chain. Here's a straight chain as well. So instead of writing the lines of the covalent bond that you see here, you see CH3, you see the C with the H and the H and the H. So you see the H here, but really it should be written in a structural way with the H's branching off the C. But you could see, you could visualize that this carbon has the hydrogens in front of it with a little 3, meaning that there's 3 hydrogens, which would be represented here. Carbons can also form a branched chain. What does this mean? This means that there could be a straight chain with a branch coming off of it. It could have more than one branch. It doesn't just have to have one branch. It could also be a ring structure. And the ring structure in this case looks like a hexagon. And you can see that some of the carbons have 2 lines, and that's what we call a double bond. So a double bond that you see between the 2 carbons, the double bond that you see here, we can see that this carbon is bonded twice to this carbon, so they're sharing even more electrons, and that's something you'll learn about in chemistry. But every carbon that we see here that I'm pointing to, these 2 carbons, have 4 bonds, and every single carbon here has 4 bonds. So let's count that up. Carbon has 1, 2, 3, 4. This carbon has 1, 2, 3, 4. And obviously this carbon has 1, 2, 3, and 4. So no matter what carbon we look at in any of these diagrams, we would see that they have 4 bonds. Each one of the carbon molecules that you see here have only carbon and only hydrogen. And because they have only carbon and only hydrogen, we call them hydrocarbons. So I'm going to write that, I'm going to write that right here. And a hydrocarbon has only carbon and only hydrogen. And so you can see clearly that all we have here are carbons and hydrogens, carbons and hydrogens, and carbons and hydrogens. This is not a hydrocarbon because it has oxygen in it. The macromolecules that we're going to talk about are carbon-based molecules. So again, we don't see the word macromolecule here just yet. So I'm going to rewrite that word up here because we are going to be talking about macromolecules. And again, macro means big, and these macromolecules are molecules found in living things. So these carbon-based molecules are oftentimes made up of several small subunits, and these are often repeating. And what we mean by that is there's several monomers linked together. And so these monomers, this word mono means one, and mer means unit. And oftentimes monomers pair with other monomers and other monomers to make a polymer. So when I look over here at our diagram, we have 1, 2, 3, 4, 5, and maybe that's not the best color. We have 5 monomers, and when I have 5 monomers together, we call it a polymer, and poly means many. And notice these are repeating units because they're all the same. They all look like hexagons. And they're all linked together by a covalent bond. So this covalent bond, again, is a special type of bond between these specific monomers. Now, this is a specific type of monomer that we're looking at. So let's take a closer look at the 4 main types of carbon-based molecules. So the 4 I've numbered here for us. We have carbohydrates, we have lipids, we have proteins, and we have nucleic acids. So we're going to start our conversation with carbohydrates. And so carbohydrates, the word refers to carbon. And then hydrate, actually, think of hydration, refers to water. And that's going to lead us to this 1-to-1 ratio. So carbohydrates are made of carbon, hydrogen, and oxygen. And the carbon, hydrogen, and oxygen that make up carbohydrates are in a special 1-to-1 ratio. That means for every 1 carbon, I have 2 hydrogens and 1 oxygen. So let's read this together. Carbohydrates are made of monomers. These monomers are called monosaccharides. So saccharide is kind of a different word for sugar. So a carbohydrate is made up of monomers called monosaccharides. These monosaccharides are going to contain the 3 elements carbon, hydrogen, and oxygen. And one of the main types of carbohydrates that we'll focus on are sugars. So let's take a look at the last couple of bullet points here. Carbohydrates can be in rings. They can also be in branches. They can have single bonds, which would be like this. They can have a double bond, which would look like this. They could have a triple bond, which would look like this. And actually, I should have made that a carbon. So they can be a little bit complex because they can be rings, branches, They can have single, double, or triple bonds. And the most simple sugar that we can talk about is called a monosaccharide. And we have an example of a monosaccharide right here. And this monosaccharide is glucose, C6H12O6. And glucose, with that formula, C6H12O6, has that same ratio, 1 to 1. So for every 1 carbon, I have 2 hydrogens and 1 oxygen. So this is the basic structure of a ring-shaped monosaccharide. So this glucose molecule that we see right here, this whole thing, this is one monosaccharide. So if I wanted to talk about the monomer of a carbohydrate, a monosaccharide would be the generic term. Glucose is an example of a monosaccharide. The representation that you see here is also an example of glucose. Notice the oxygen, and there's the oxygen here. So there's a difference in the way this one is drawn versus this example. This is a simplified version, and we actually call glucose a hexose sugar. And obviously it's hexose because it has like a 6-sided hexagon shape. And anytime you see OSE, you should think sugar. So OSE, sugar. So up here, back to this example again at the top, if we were to look at this, we can see we have a polymer. It's made up of 1, 2, 3, 4, 5 hexose sugars. Each one we could call a monomer, or each one we could call a monosaccharide. And together, the entire thing would be a polysaccharide. So we're still talking about carbohydrates. And carbohydrates, again, are one of the 4 macromolecules. And carbohydrates can be broken down to provide energy for our cells. There's energy between the carbon-carbon bonds. So there's stored energy here. And when we break those bonds, we release energy. Carbohydrates can also be part of the cell structure, and we're going to talk about that when we get into our discussion of the cell. Now, as I mentioned before, we can have monosaccharides or polysaccharides. There are 3 examples of polysaccharides that you should be familiar with: starches, cellulose, and glycogen. Now, there's something really interesting about these 3. All 3 of them are polysaccharides, which it says that in the text that you just saw, but all 3 of them are made of glucose monomers. So I'm going to kind of write that over to the side here. All 3 are made of glucose monomers. So what makes them different from each other? Because they are different from each other. They're all made up of glucose, but it's how the glucose molecules are put together that makes them different. Where do we find starches? Well, in plants, plants store, and actually, let me rewrite that. Plants store glucose as starch. Animals store glucose as glycogen. So you and I when we eat and we store the energy from the food that we eat, we store that energy as glycogen. But plants store that energy as starch. So remember what we said, carbon-carbon bonds have stored energy. Now, I didn't mention cellulose yet. Plants have cellulose in their cell walls. Oops. So now we've talked about 3 polysaccharides, and these 3 polysaccharides are made up of one type of monomer, and that monomer is glucose. So when we talk about glucose, it can be stored as different things. And we should probably add here, plants have cellulose in their cell walls, and cellulose is made of, you guessed it, glucose. So let's take a closer look at the structure of a carbohydrate. So remember, we've only been talking about carbohydrates. Carbohydrates can be referred to as sugars and starches. We know that carbohydrates are made up of carbon, hydrogen, and oxygen. Carbohydrates can be monosaccharides or polysaccharides. We could even say a disaccharide, which would be 2. So this structure right here is starch. And here you see cellulose. Now they kind of look the same, but what's different? There's a branch right there. So starch is more of a branched molecule, where cellulose is more of a linear molecule. But if you notice, each one of these monomers looks the same because they're all glucose. Every single monomer that you see here is glucose. So let's look at what it looks like in a more complicated way. So over here, you see all of the carbons, hydrogens, and oxygens. Now, each one of these is also glucose. So I have several monomers here linked together. So here are my covalent bonds. And in these covalent bonds, I'm linking monomers together to create a polymer. Our next macromolecule is the lipid. Notice it says straight away that lipids are nonpolar. We know what nonpolar means. Nonpolar means that there is no charge. So there's no attraction to anything. Lipids in general include fats, oils, cholesterol. We could even say waxes are included in lipids. Lipids are not true polymers. Now remember, what makes a polymer? A polymer is created from one monomer added to another, to another, to another. Lipids are not a bunch of monomers added together. So they are macromolecules, which means they're large molecules, but they're not polymers. So let's look at the chemical makeup of a lipid. So lipids contain carbon chains called fatty acids. So here's my carbon chain. This would be my fatty acid. And notice I don't just have one, I have 2, 3 fatty acid chains or tails. And some people call them tails. So fats and oils and waxes have these fatty acids that we just looked at here. They have fatty acids, but these fatty acids are always linked to something we call a glycerol, which is over here. So when we look at a lipid, it's important to remember it's important to remember that we're looking for some basic components, just like we looked for basic components in a carbohydrate. So in a carbohydrate, we looked for carbon, hydrogen, and oxygen in a 1-to-1 ratio. Here it's going to be a little different. We're going to look for carbon, hydrogen, and oxygen, but this time the ratio is not 1-to-1. It's 1 to very few. So when I look at my carbons, for every one carbon, I have 2 hydrogens bonded here. So I can kind of eyeball that and see that for every one carbon, I have 2 hydrogens. But there's 0 oxygens here. The only place I see oxygens are in this component of the molecule. And this portion of the molecule, again, is called the glycerol. So we have our glycerol and we have our fatty acid tails or chains. So what's the function of a lipid? Now, lipids can be specialized and have different functions, but some of the main functions that they have, remember, they're fats, so they can be a source of energy. This is more like a long-term source of energy. Carbohydrates are like short-term energy. They also make up cell membranes, which we're going to look at in a future slide. That's about 2 slides down, I believe. And they're also used to make hormones like estrogen and testosterone and cholesterol. We'll be looking at each of these as a function of what lipids do for us. So let's first talk about lipids as a source of energy. So lipids as a source of energy, if we think about what we said about energy in a carbohydrate, there's lots of energy between a carbon-carbon bond. Well, when I look at my lipid, I like this diagram a little bit better because of the way they've drawn it. When I look at my lipid, I have lots of carbon-carbon bonds. So there's lots of energy stored in those carbon-carbon bonds. Now, lipids can be classified, the ones that I'm talking about that we use for energy, as fats and oils. They can be saturated or unsaturated. And you've probably heard that before, like saturated fats, unsaturated fats. Saturated fats are bad for you, unsaturated fats are better for you, and things like that. So we're going to talk about that just a little bit. Saturated fats typically come from animal products, and unsaturated fats typically come from plant products or plants, like if we eat avocados or walnuts or any kind of nut really, or anything, any plant that's considered to have fats like olive oil, that would be considered an unsaturated fat as well. So why do we give them the names saturated and unsaturated? So let's look over here at what it says. Saturated fats contain fatty acid tails or fatty acids in which all carbon-carbon bonds are single bonds. So when I look at this fatty acid tail, I see only single bonds. That means one line. So that's one covalent bond between each carbon. Now, what I don't like about this diagram, and you might find confusing, is the way that they've drawn the hydrogens. So we just have to imagine that each one of these hydrogens there would be one here and one here. That's where these 2 hydrogens would be. They've just written it out this way instead of writing it out structurally the way that I just did. So what is an unsaturated fat then? If saturated fats have all single bonds between the carbons, what is an unsaturated fat? There has to be at least one carbon-carbon double bond, just one. When I look at this fatty acid tail, I have single, 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 single. I have a double, I have a double. I have more than one, so it has to be unsaturated. Even if I only had one, it would be, if I had one double bond, it would be unsaturated. So I have a double bond, a double bond, a double bond, and a double bond. So I have several double bonds, so this is definitely unsaturated. Now I want you to take a look at this carbon here. There's 2 double bonds, and carbon only needs 4 bonds, if we remember back to what we started with. So 1, 2, 3, 4. It doesn't need any hydrogen, so you don't see any H's around it. This carbon it has two bonds with this carbon. So it has one double bond here. It has a single bond with this carbon, and then it has another hydrogen. So if we were to draw this the way the other diagram looks, then we would have the hydrogen here. And it wouldn't be right there. So what does this mean? How does this look physically to the lipid? The lipid will look like a solid if it is saturated because of all the single bonds. It will appear to us as a liquid if it has double bonds and it is unsaturated. So we would have to have one or more double bonds to be liquefied. And you may be familiar with coconut oil. Coconut oil can be kind of semi-solid in the winter and a little bit more liquidy as the temperature gets warmer. So just to kind of bring back a point we mentioned earlier, this would be considered my fatty acid tail here, and then this would be considered my glycerol. So here, this would be my glycerol, and this would be my fatty acid tail. And this would be my unsaturated fatty acid tail. And here would be my saturated fatty acid. So again, we have that ratio 1 to very few. So for every carbon, I have 2 hydrogens just about. Now that's a little different here. But I have very few oxygens compared to how many carbons and hydrogens I have. So 1 to very few. Let's take a look at lipids in our cell membrane. So many of you've heard of the idea of a cell membrane before. And cell membranes have a specific composition or something specific that makes them up. And what makes them up is this structure called a phospholipid. And a phospholipid's kind of interesting because it is a lipid, and because it's a lipid, it has carbon, hydrogen, and oxygen. 1, 2, very few. And that would be in this portion of the molecule. So notice it has 2 fatty acid tails. So this is one phospholipid, and it has 2 fatty acid tails, but it has this phosphate head. So this phosphate head makes this lipid unique. And it makes it unique because it's a lipid that has phosphorus. And there's the phosphorus. And notice it's negatively charged. So what did we say about lipids earlier? We said that lipids in general are nonpolar. But what part of the molecule is nonpolar? It's really this part of the molecule that's nonpolar, which is the fatty acid tail, which is only carbon and hydrogen, which earlier we said was nonpolar. But we have this phosphate head, and this phosphate head has a charge. And that charge is negative. And we know that charged molecules are polar. So the head is polar, but the tail is nonpolar. So we have a polar head and we have a nonpolar tail. Polar head, nonpolar tail. So I've zoomed into a layer of the cell membrane, and this layer of the cell membrane has phospholipids. And these phospholipids, they look a little different here because they're a little bit, I don't know, bigger, funkier. You can see them. And the arrangement is that we have a polar head facing this way with our nonpolar tails, and then we have another layer of nonpolar tails with our polar head. And so this portion is nonpolar, and this is outside of the cell. So out here, all of these heads are touching the outside of the cell. This is external of the cell on this side. So that means that in here, this would be internal of the cell. So these are two different environments. This is what's inside the cell. This is what's outside the cell. Well, there's one thing that the inside of the cell and the outside of the cell have in common. And that is the inside of the cell and the outside of the cell both are surrounded by water. And what do we know about water? Water is polar. And what do we know about the phospholipid? It has a polar head. It has charges. Those charges are attracted to the water molecules. So the polar heads arrange themselves so they're sticking out towards the water, or they're sticking in towards the water, and the tails are on the inside. And right here we have a bunch of tails facing each other. So it's interesting to see that the tails are together, but the heads are facing the water. And hopefully that makes sense because the tails aren't even attracted to the water. So the heads are. And wherever the head goes, the tails just follow. So when the heads rearrange themselves toward the water molecules, the tails are on the other side, so they end up facing each other. So one last thing that I'm going to say about the cell membrane, and that is the fact that it doesn't have one phospholipid layer, it has two. So we would say this is a layer of phospholipids, and this is a layer of phospholipids. So I have two layers of phospholipids, and we call those bilayers. Now, the other thing that we want to say about the cell membrane is there are other molecules inside of it. There are proteins inside of it. There are carbohydrates sticking out of it, and there's cholesterol in it. So let's take a look at what's what. So our proteins are here. You can see it in yellow. There's a protein. Here's another protein. So I have two proteins embedded in this membrane. I have proteins out here sticking out. And this shape should look familiar because there's lots of hexagons, which looks like a carbohydrate or a bunch of glucose molecules. And then last but not least, we have our cholesterol, which is here. And cholesterol is unique because it has 4 fused rings. So you can see here, 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. These are all cholesterol. And so the difference between knowing it's cholesterol and a carbohydrate is the fact that the rings are fused. And by that we mean the rings are butted up against each other. Sorry, my rings aren't very good. That's supposed to be a hexagon. So they're fused right here. So this would be like a cholesterol. I have to draw another one to make it cholesterol because it needs to be 4. So 1, 2, 3, 4 fused rings, which is very different from a carbohydrate, because a carbohydrate, it wouldn't be fused. Let's go back and back. Sorry about this. Notice that these rings are not fused. There's a bond between them, so they're not right next to each other. So a fused ring, again, has to be like this. So they're fused right here. And again, my rings are not, they're not very symmetrical, but these are not fused. And these are. So that sums up our conversation about carbohydrates and lipids. We will talk about the other 2 macromolecules in another session. And I hope this was helpful.